Somewhere in the North Atlantic, a 50-ton animal is hanging in the dark, head pointed straight down, body completely still, eyes closed. It is not surfacing, it is not hunting, it is not doing anything we can name with confidence. It has been like this for minutes, and we have no idea why. Sperm whales can hold their breath for over an hour. They dive to depths of more than a mile, deeper than most submarines are designed to go. They have been doing this for millions of years, across every ocean on the planet. We have studied them for centuries, and we still cannot explain some of the most basic things about them. Inside their skull sits an organ unlike anything else in nature, a massive structure filled with a waxy, oily fluid, taking up a huge portion of the entire head. It has been examined, debated, and written about for hundreds of years. Scientists still do not agree on what it does. They carry the largest brain of any animal that has ever lived on this planet, up to 20 pounds of neural tissue, with internal structures found in no other species. They produce the loudest sound ever recorded from a living creature, a click so powerful it can be felt, not just heard. And across oceans, separated by thousands of miles of open water, different family groups produce different patterns of clicks. The same patterns passed from mothers to daughters, generation after generation, like a language handed down through time. Researchers have been recording those patterns for decades. None of it has been decoded. This is one of the largest, loudest, most physically extraordinary animals in the history of life on Earth. We have entire research programs dedicated to understanding it. We have decades of recordings, decades of dive data, decades of observation, and it is still, in fundamental ways, a mystery. Today we go through what we know, what we don't, and why the gap between those two things is so much wider than most people realize. Now let's go deeper, long before any vessel crossed the open ocean, before charts traced coastlines or instruments measured depth. A lineage was already moving through water with a purpose shaped across immense spans of time. The animal that exists now carries that continuity within its form, not as a sudden creation, but as the result of a gradual return, a reversal written into bone and motion. Its presence feels complete, yet its origin begins far from the sea. Sperm whales, known scientifically as Physeter macrocephalus, are the largest toothed predators ever to exist, with adult males reaching lengths of 67 feet and weights approaching 57 tons. Their scale alone places them outside familiar comparisons, not only larger than any living terrestrial predator, but structured for a world where size behaves differently under pressure and depth. Despite their immense bodies, they do not belong to the same lineage as the baleen whales that filter feed in shallower waters. Their evolutionary path diverged roughly 30 million years ago, forming a separate branch defined not by passive feeding, but by pursuit, precision, and sound. The earliest ancestors of this lineage did not resemble what moves through the ocean today. Fossil records reveal creatures with legs, with articulated hips, with weight-bearing limbs that once pressed against solid ground. These early cetaceans lived at the boundary between land and water, gradually shifting their bodies toward buoyancy and propulsion. Over approximately 15 million years, their forms reshaped themselves, limbs shortening, spines flexing, skulls elongating, until movement through water became not an adaptation, but a default state. This transformation remains one of the clearest evolutionary reversals preserved in the fossil record, a slow migration away from land into a medium that would eventually define every aspect of their biology. Now, they inhabit every major ocean basin on Earth, from equatorial regions where sunlight saturates the surface, to the cold margins near polar ice, where light fades quickly into gray. Their distribution is vast, yet their lives are structured in ways that remain intimate and precise. Female sperm whales live within stable, multi-generational family units, typically composed of 10 to 12 individuals. These groups persist for decades, maintaining relationships that extend across entire lifespans. Within them, movement, communication, and survival are shared responsibilities, carried collectively rather than individually. Male sperm whales follow a different path. After adolescence, they leave these family units and spend much of their lives alone or in loose associations with other males. Their trajectories take them into colder, higher latitude waters, where they grow larger over time, eventually returning toward warmer regions, only for breeding. This separation creates two parallel lives within the same species, one deeply social and continuous, the other distant and solitary, both shaped by the same underlying biology, yet expressed through different patterns of existence. The global population today is estimated between 300,000 and 450,000 individuals, a number that reflects both resilience and loss. Before the era of industrial whaling, their numbers were closer to 1.1 million. Within less than 2 centuries, that population was reduced by nearly 3/4. The scale of that reduction exists not only in numbers, but in the disruption of structures that take generations to form. Even as populations recover slowly, the absence of what was removed remains embedded in the present. A sperm whale can live up to 70 years, carrying with it decades of accumulated experience, social connection, and environmental memory. These long lifespans allow knowledge to persist within groups, passed not through instinct alone, but through repeated interaction and shared behavior. And yet, despite centuries of hunting, observation, and scientific study, the fundamental nature of this animal remains unresolved. It has been measured, tracked, recorded, and still exists partially outside the frameworks built to understand it, moving through an environment that conceals as much as it reveals, leaving behind a presence that is unmistakable but never fully explained. It begins as pressure, not as sound, a brief compression moving through water before any ear could register it as anything at all. Within that instant, energy is released with a precision that feels almost mechanical, shaped and directed long before it leaves the body that produced it. By the time it travels outward, it has already been refined into one of the most intense biological signals ever recorded. A sperm whale click reaches 230 decibels, a value that only gains meaning when placed against something familiar. A jet engine at 100 feet produces around 140 decibels, already strong enough to overwhelm human hearing. The decibel scale does not rise in simple steps, but in exponential leaps, each increase of 10 representing a tenfold growth in intensity. At 230, the energy contained within a single click moves beyond loudness into force, capable of interacting physically with matter in its path. This sound is not formed by vocal cords, as there are none present. Instead, the process unfolds within the whale's head, inside a system of air-filled passages and dense, specialized tissues. Air is driven through a structure near the front of the skull known as the museau du singe, sometimes called the monkey's lips, where it produces an initial pulse. That pulse does not simply exit the body, it reflects backward against the inner surfaces of the skull, then passes through layers of fatty material before being directed forward again. The spermaceti organ, embedded within this system, acts as a shaping medium, focusing the energy into a narrow, forward-facing beam. What emerges is a click lasting less than 1 millisecond, a duration so brief it exists almost outside ordinary perception. Yet within that fraction of time, there are layers, subtle variations in structure that can be measured and distinguished. The sound travels through seawater at roughly 1,500 meters per second, spreading outward until it encounters objects in the environment, then returning as echoes that carry information about distance, size, and texture. Hydrophones placed miles away can detect these signals, tracing the presence of an animal that remains unseen. For the whale, this system functions as a way of perceiving space where light does not reach. A squid the size of a football, suspended at 1,000 feet, becomes detectable, not through sight but through the returning pattern of reflected sound. Each click sends out a brief map, and each echo fills in another part of a three-dimensional understanding that is constantly updating. Movement, shape, and position emerge from these returning signals, forming a sensory field that surrounds the animal as it moves. At close range, the energy contained within these clicks becomes more than informational. The same focused pulse that reveals a target may also disrupt it, delivering enough force to disorient or incapacitate prey. This dual capacity, perception, and interaction within a single signal suggests a system that does not separate sensing from acting, but combines them into a single process. And within this system lies an unresolved boundary. The clicks used for navigation and hunting are also used in sequences exchanged between individuals, structured patterns that repeat and vary across groups. The mechanism that produces them remains the same, the pathways identical, the structures unchanged. Yet the purpose appears to shift from mapping the external world to shaping interactions within a social one. How that transition occurs is not directly visible. There is no clear signal that marks the change, no observable switch that separates one function from the other. The same pulse moves outward, carrying with it either a reflection of the environment or something shared between minds, and from the outside both appear as the same brief, concentrated release of sound. Across long recordings taken far from shore, the sound begins to organize itself into patterns that do not belong to hunting or navigation. The intervals settle into rhythms, pulses spaced with a consistency that suggests intention rather than reflection. These sequences, known as Kodas, unfold as brief arrangements of clicks, repeating in recognizable forms that carry structure without revealing their meaning. Unlike the isolated pulses used to sense the surroundings, Kodas arrive as groups, each click placed in relation to the next. The spacing can be even or irregular, tight clusters or stretched intervals, forming signatures that can be identified and cataloged. They are not random emissions and not simple echoes returning from distant objects. They are produced in sequences that remain stable across time, as if each arrangement holds a specific place within a larger system that has yet to be fully understood. The first detailed descriptions of these patterns emerged in the 1980s, when researcher Catherine Payne and her colleagues began analyzing extended acoustic recordings. What appeared at first as scattered noise resolved into repeating structures, each one distinct enough to be recognized again when it occurred. As more data accumulated, these patterns multiplied. In the Pacific alone, more than 21 separate coda types have been documented, each defined by its rhythm and internal spacing rather than by pitch or tone. These codas are not shared uniformly across all sperm whales. Different family groups maintain their own repertoires, sets of patterns that remain consistent within the group and distinct from those of others. A family living near the Galapagos Islands produces sequences that differ from those recorded in the Caribbean, and these differences persist across years, even as individuals move and generations change. The patterns remain tied to social units, suggesting a system that is maintained through interaction rather than inherited fully formed. Young whales do not begin life with this system already in place. Calves produce early sequences that lack precision, uneven in timing, and inconsistent in structure. Over time, through repeated exposure to the patterns used by their family, these early attempts begin to align more closely with established forms. The spacing becomes more regular, the sequences more recognizable, until the calves' contributions blend into the shared repertoire of the group. This gradual refinement mirrors processes seen in other forms of learned communication, where repetition and feedback shape the final structure. Among all known Kodas, one stands apart in its distribution. The sequence referred to as the 5 regular consists of 5 evenly spaced clicks, simple in form yet present across nearly every studied population worldwide. It appears in groups that otherwise share no overlap in their repertoires, a common pattern embedded within a system defined by variation. Its persistence across geographic boundaries suggests a function that remains constant, though what that function represents is not yet clear. Efforts to interpret these sequences have expanded beyond traditional observation. Project CT, the Cetacean Translation Initiative, has begun applying machine learning techniques to vast collections of recorded Kodas. Thousands of hours of data are analyzed, searching for recurring structures, correlations with behavior, and patterns that might indicate the presence of grammar or syntax. The approach assumes that meaning, if present, may emerge through scale, through the accumulation of enough examples to reveal underlying rules. As of 2024, no coda has been definitively decoded. The sequences remain structured yet opaque, carrying information that has not been translated into human terms. It is not known whether they convey identity, emotional state, coordination, or concepts that have no direct parallel in human language. The patterns continue, repeating through the water, consistent and precise, forming a system that exists clearly enough to be recorded, yet remains beyond interpretation. Inside the massive architecture of the sperm whale's head lies a volume so large that early anatomists, working without context, believed they had found the seat of its intelligence. What they had opened instead was something else entirely, a structure occupying nearly 1/3 of the animal's total length, extending forward through the skull as a dense continuous mass. It does not resemble any familiar organ, neither in form nor in composition, and even now it resists simple classification. This structure is known as the spermaceti organ. It is filled with a pale, wax-like substance, translucent when warmed, solidifying as it cools, a material unlike typical biological fluids. During the 18th and 19th centuries, this substance became one of the primary reasons sperm whales were hunted. It burned cleanly in lamps, produced little smoke, and could be refined into lubricants and cosmetics. Historical accounts note its use in maintaining the rigging of naval ships, including the USS Constitution, where its stability under varying temperatures made it particularly valuable. The material's properties were well understood long before its purpose within the living animal was. Within the whale, the spermaceti organ is integrated into a broader system of tissues and air spaces that shape and direct sound. One prevailing hypothesis suggests that it functions as an acoustic lens, focusing the energy generated within the head into a controlled forward-moving beam. The density and arrangement of the spermaceti material allow it to refract and guide sound waves, aligning them into a coherent path. This interpretation fits with the precision of the whale's echolocation, where returning echoes provide detailed spatial information in environments where light cannot penetrate. Another possibility considers the physical properties of the spermaceti itself. As temperature changes, the substance shifts in density, becoming slightly more solid as it cools, and more fluid as it warms. Some researchers have proposed that this change could influence buoyancy, allowing the whale to adjust its position in the water column with minimal muscular effort. By cooling the spermaceti during descent and warming it during ascent, the whale might achieve subtle control over its vertical movement, functioning as a kind of internal ballast system that operates without external motion. A third interpretation focuses on the concentration of energy within the click system. The spermaceti organ may act not only to shape sound, but to intensify it, directing acoustic force toward nearby prey. At close range, this concentrated energy could disorient or incapacitate, turning a sensory mechanism into an active tool within the act of feeding. The structure of the organ, combined with the surrounding tissues, supports the possibility of such amplification, though direct observation of this effect remains limited. Each of these hypotheses is supported by partial evidence, drawn from anatomical study, physical modeling, and indirect observation. None has been confirmed in isolation, and it is increasingly considered that the organ may perform several roles simultaneously. Its structure allows for multiple interactions with sound and movement, yet the way these functions are coordinated within the living animal remains unclear. There is no direct method to observe the organ in operation under natural conditions. The difficulty lies in access. The spermaceti organ is positioned deep within the skull, surrounded by dense bone and sensitive tissues. Studying it in a living whale without causing harm is not currently possible. As a result, nearly all detailed knowledge comes from examinations of stranded individuals, animals whose physiological states may differ significantly from those in the open ocean. What is observed in these cases provides valuable insight, but it represents only a partial view, shaped by circumstances that do not reflect the full range of natural behavior. Within this quiet absence of direct observation, the organ remains defined more by inference than certainty, a structure that is clearly central to the whale's existence, yet still not fully understood in its purpose. The descent begins without urgency, a steady surrender to depth, measured not in distance alone, but in changing conditions that gather around the body. At roughly 6 feet per second, the whale moves downward through layers that gradually remove the last traces of surface light, each moment carrying it further from any visual reference. Over the course of 30 to 45 minutes, it reaches depths of 3,000 feet or more, entering a region where illumination no longer exists and orientation depends entirely on other senses. At that depth, pressure builds to over 1,300 pounds per square inch, a force capable of deforming rigid metal structures. The whale's body does not resist this pressure in the way engineered vessels attempt to. Instead, it yields in controlled ways. The rib cage is only partially fixed to the sternum, allowing the chest to compress as the surrounding water presses inward. The lungs collapse safely, reducing the risk of nitrogen absorption and structural damage, while other tissues accommodate the shifting volume without disruption. Oxygen becomes a finite resource that must be managed with precision. The spleen, enlarged beyond what is typical in most mammals, serves as a reserve, storing concentrated red blood cells that are released gradually during the dive. As depth increases, this reservoir supplements the oxygen already carried in the blood, extending the time the whale can remain submerged. This process is not unique to sperm whales, but within them it is amplified, scaled to match the demands of prolonged descent. Circulation changes as the dive deepens. Blood flow is redirected away from organs that are not immediately essential, concentrating instead on the brain and the heart. This redistribution, known as the diving response, ensures that critical systems remain supplied even as overall oxygen levels decline. At the same time, the heart rate slows significantly, dropping to as few as 4 beats per minute during the deepest phases. This reduction in activity lowers metabolic demand, stretching the available oxygen across a longer period. In the absence of light, the environment offers no visual boundaries, no contrast, no horizon. Yet the whale continues with purpose, navigating and locating prey within this space. Giant squid inhabit these depths, organisms that can reach lengths exceeding 40 feet, equipped with tentacles lined with suction cups that grip with considerable force. Encounters between whale and squid unfold beyond direct observation, leaving only indirect evidence. Circular scars, some more than 4 inches across, mark the skin of adult whales, patterns that correspond to the dimensions of squid suckers. These markings persist as records of contact, each one indicating a moment of resistance within the darkness. Feeding occurs within this same environment, where movement is guided by non-visual perception and interaction is immediate. The whale captures its prey and consumes it at depth, sustaining itself on organisms that rarely approach the surface. The act is efficient, repeated across dives that follow similar trajectories, each descent shaped by the same physiological constraints and adaptations. After this period at depth, the ascent begins. The whale rises gradually, reversing the path taken downward, though not always in a continuous motion. At around 1,000 feet, pauses are often observed, intervals where vertical movement slows or stops for a time. The function of these pauses has not been clearly determined. They may relate to physiological adjustment, to digestion, or to processes that remain unmeasured. From the outside, the dive appears as a simple disappearance followed by a return. Within it, a sequence of controlled changes unfolds, each one allowing the animal to move through conditions that would otherwise be inaccessible. The descent, the stillness at depth, and the measured ascent form a cycle repeated throughout its life, a passage through darkness that remains largely unseen, defined by mechanisms that continue to be studied in fragments rather than as a complete, observable whole. For centuries, the presence of something vast and soft-bodied, moving far below the reach of light, was treated as rumor more than record, a shape suggested by damaged nets and fragments brought to the surface rather than by direct sight. Only in 2004 did a living giant squid, Architeuthis dux, appear clearly within its own environment, captured not as a legend but as an animal, suspended in its native depth. Even then, the image arrived as a brief confirmation of existence rather than a full understanding of its life. The scale of this organism places it among the largest invertebrates on Earth. Females average around 33 feet in length, males somewhat smaller at roughly 26 feet, though individuals have been recorded reaching up to 43 feet. These measurements include the long feeding tentacles that extend well beyond the main body, giving the animal a reach that exceeds its core mass. Its form is elongated, streamlined for slow movement through dense water, with a mantle that encloses its organs and fins that provide subtle directional control. It lives within the same depth range that sperm whales enter during their foraging dives, generally between 1,000 and 3,300 feet. This overlap places both species within a shared zone where light does not penetrate and where encounters occur beyond any direct human observation. Within this environment, the squid relies on sensory adaptations that differ from those of its predator. Its eyes are the largest known in the animal kingdom, measuring up to 10.6 inches in diameter. These structures are capable of detecting faint changes in the surrounding darkness, including the slight disturbances in bioluminescence created by movement through the water. The interaction between sperm whales and giant squid has been reconstructed almost entirely from indirect evidence. Inside the stomachs of whales, researchers have found the remains of squid in the form of beaks, rigid structures that resist digestion. In some cases, a single whale has contained hundreds of these beaks, accumulated over time. One documented stomach held more than 18,000 beaks, representing an extended history of feeding rather than a single event. Each beak corresponds to an individual squid, a record preserved not in observation, but in residue. On the surface of the whale's body, additional traces appear. Circular scars, often arranged in patterns along the head and flanks, match the dimensions and spacing of squid suction cups. Some of these marks exceed 4 inches across, indicating contact with large individuals capable of exerting considerable force. These impressions remain long after the encounter, a physical archive of interactions that took place in conditions where no visual record exists. The process by which the whale captures the squid remains unresolved. The environment in which these events occur prevents direct study, and no complete observation of a successful hunt has been recorded. It is possible that the whale uses its acoustic system to disorient prey at close range, or that it relies on rapid approach guided by non-visual cues. Other interpretations suggest ambush, or a combination of movement and sensing that has not yet been fully described. Each possibility aligns with aspects of known anatomy and behavior, yet none can be confirmed through direct evidence. The only recorded footage of such an encounter shows a whale approaching a squid at depth. The squid, detecting the disturbance, extends its tentacles and moves away, escaping the immediate reach of the predator. The sequence ends without resolution, leaving the outcome uncertain. From this single observation, no consistent pattern of success or failure can be established. What remains is a relationship defined by fragments, by remnants recovered and marks observed, by brief glimpses that suggest interaction without revealing its full form. The whale descends repeatedly into this shared space, and the squid continues to inhabit it, both moving through the same layer of the ocean where contact occurs without witness. Near the surface, where the water softens and light returns in muted layers, there are moments when movement ceases entirely. A group gathers without sound or visible coordination, bodies aligning into a shared stillness that does not resemble any familiar form of rest. Each animal hangs suspended, head angled upward, tail descending into deeper water, as if anchored between two worlds without touching either. This arrangement was first recorded in 2008 off the coast of Chile, when researchers encountered a cluster of sperm whales drifting in this upright posture. There was no sign of distress or active behavior, only a quiet suspension that persisted as the boat approached. The animals remained unresponsive, their bodies steady, their orientation unchanged. Over time, further observations across multiple ocean basins confirmed that this was not an isolated event, but a recurring behavior present throughout the species. In this state, sperm whales sleep. They remain vertical, often in tight formations, holding this position for intervals that typically last between 10 and 15 minutes. During these periods, there is no visible reaction to nearby movement. Boats can pass within close range, and the whales do not shift or scatter. The stillness is complete, a temporary withdrawal that contrasts with the otherwise continuous motion of life at sea. Across an entire day, the total time spent in this state is remarkably brief. Sperm whales sleep for approximately 7% of a 24-hour period, amounting to roughly 1.7 hours in total. This is the lowest recorded sleep duration among mammals, a pattern shaped by the demands of an environment where breathing requires conscious surfacing and extended absence carries risk. Sleep occurs in short segments, distributed rather than continuous, woven into the intervals between other behaviors. In most marine mammals, rest is achieved through a process known as unihemispheric sleep. One half of the brain enters a resting state while the other remains active, maintaining control over breathing and awareness of surroundings. Dolphins, for example, alternate between hemispheres, allowing them to remain partially alert even while resting. This mechanism ensures that essential functions continue uninterrupted. Sperm whales, however, appear to diverge from this pattern. Observations suggest that both hemispheres of the brain may enter a sleep state simultaneously during these vertical rest periods. The body remains upright, the surface close, yet the usual safeguards associated with unihemispheric sleep do not seem to apply in the same way. How breathing is maintained under these conditions, how the transition between sleep and wakefulness is managed, remains uncertain. The behavior persists without a clear neurological explanation. The vertical posture itself introduces another layer of ambiguity. One possibility is that this orientation allows the blowhole to remain near the surface without active movement, reducing the need for muscular effort during rest. Another interpretation considers the arrangement of the group, where vertical alignment creates a more compact formation, maintaining proximity between individuals in a space where horizontal distance could quickly expand. Both ideas align with aspects of the observed behavior, yet neither has been confirmed as a definitive explanation. What remains visible is the stillness itself, a pause that interrupts the otherwise continuous motion of these animals. Suspended in the water, they occupy a position that does not correspond to typical patterns of rest seen in other species. The body remains poised, neither drifting horizontally nor sinking, held in place by forces that are not immediately apparent. For brief intervals, the activity of the animal subsides into this quiet alignment, then gradually returns, the group dispersing as movement resumes. Within these moments, the boundary between activity and rest becomes less distinct, shaped by conditions that do not allow for complete disengagement. The whales sleep, but in a form that remains partially unexplained, defined by posture, duration, and a neurological pattern that has yet to be fully understood. Within certain groups, movement does not emerge from consensus or chance, but from a single point of continuity carried quietly at the center. The formation holds together across distance and time, not through visible signals, but through an order that remains consistent even as individuals change. At the core of this structure is an individual whose influence is not defined by size or force, but by duration. Sperm whale society is organized around matrilineal units, among the most stable social systems observed in any marine species. Each group is led by the oldest female, the matriarch, whose age can be traced through growth layers embedded in her teeth, accumulating year by year in a pattern similar to rings within a tree. Some individuals have been estimated to exceed 60 years, their lives extending across decades of shifting conditions. Within that span, experience accumulates in ways that cannot be reduced to instinct alone. The matriarch's role is not expressed through dominance in the physical sense, but through the retention and application of knowledge. She carries with her an internal record of movement through the ocean, a familiarity with regions where food appears and disappears with seasonal cycles, an awareness of areas where threats are more likely to occur, and an understanding of subtle environmental changes that influence the behavior of prey. These patterns are not static, they shift gradually, and the ability to track those changes over long periods becomes a central resource for the group. A parallel can be found on land within the social structure of African elephant herds, where older females guide the group through periods of scarcity. Studies have shown that herds led by older matriarchs have higher survival rates during drought conditions, as these individuals remember the locations of water sources that have not been used in decades. The relevance of this comparison lies not in direct similarity, but in the shared reliance on memory as a survival mechanism, where information accumulated over long lifespans becomes essential in navigating uncertain environments. In sperm whales, the same principle is strongly suggested, though it has not been directly measured with the same clarity. Tracking individual whales across multiple decades presents significant challenges, as consistent observation requires technologies that have only recently begun to develop. Tagging systems can follow movement for limited periods, but they do not yet provide a complete record of how knowledge is applied or transferred within the group over time. What can be observed instead are patterns that emerge when this continuity is disrupted. During the period of intensive commercial whaling, large numbers of mature individuals were removed from populations, including many matriarchs. In the aftermath, some family units exhibited changes in behavior that indicated a loss of internal structure. Movements became less coordinated, foraging patterns less efficient, and instances of strandings appeared more frequently. These shifts suggest that the absence of experienced individuals alters the way groups interact with their environment, though the exact mechanisms remain difficult to isolate. Beyond navigation, the transmission of knowledge extends into other aspects of life. The distinct coda patterns used within each group are not inherited as fixed signals, but learned over time, refined through repeated interaction. Foraging strategies, responses to external disturbances such as vessel, noise, and patterns of association within the group all show signs of being shaped through experience rather than encoded entirely in biology. The matriarch, by virtue of her longevity, becomes a central point through which these patterns persist. Within this structure, survival is not solely a matter of individual capability, but of continuity across generations. The group functions as a repository of accumulated experience, and the matriarch serves as its most enduring link, holding together patterns that extend beyond any single lifetime. At the surface, where the water remains within reach of air, there are intervals when the youngest members of the group do not follow the others below. Their bodies are still developing, their systems not yet prepared for the pressures that define the deeper layers. While the adults descend in measured sequences, the calves remain above, held within a different rhythm, one that depends on proximity and protection rather than movement through depth. A sperm whale calf cannot accompany its mother on extended foraging dives. The physiological adjustments required to tolerate compression, reduced oxygen availability, and prolonged submersion have not yet matured. Its lungs, circulation, and internal reserves are still adapting to the demands of life at sea. As a result, when the mother begins her descent, the calf stays behind, suspended in a region where exposure becomes a different kind of risk. At the surface, vulnerability shifts from internal limitation to external threat. Sharks and orcas move through these waters, and a calf alone would present an opportunity, yet isolation does not occur. Instead, other females within the family group remain alongside the calf, forming a presence that replaces absence with continuity. These individuals are not always the calf's mother. They are relatives, companions, members of the same social unit, and their role extends beyond observation. This system is known as allomothering. While one female descends to forage, others remain above, maintaining contact with the calf and providing care that mirrors maternal behavior. They position themselves nearby, adjust their movement to match the calf's, and allow physical closeness to persist during the mother's absence. In some cases, this care includes nursing. Calves have been observed feeding from multiple females within the same group, sometimes receiving milk from as many as 5 different individuals over a given period. The production of milk in these non-mothers introduces a process that is not fully understood. Hormonal changes appear to be influenced by social context rather than direct reproduction, suggesting that the presence of a calf and the structure of the group can initiate physiological responses. The mechanisms behind this response remain only partially described, and the conditions under which it occurs are still being studied. From an energetic perspective, this behavior carries a cost. Producing milk requires substantial metabolic resources, and in many species, such investment is limited to direct offspring. Evolutionary models would predict that providing nourishment to unrelated young would reduce individual fitness, as energy is diverted away from personal reproductive success. Yet in sperm whales, allomothering is consistent across observed populations, indicating that the behavior persists despite this apparent contradiction. One explanation centers on the genetic relationships within the group. Females in a family unit are often closely related, sharing portions of their genetic material. By supporting the survival of a calf within the group, even if it is not their own, they contribute indirectly to the continuation of shared genes. This concept, known as kin selection, provides a framework in which cooperative care becomes advantageous at the level of the group rather than the individual. However, the exact degree of relatedness required to offset the energetic cost has not been clearly established through genetic analysis. Studies have yet to determine whether the observed behavior aligns precisely with predicted thresholds or whether additional factors influence its persistence. Social cohesion, mutual dependence, and the stability of the group may also play roles that extend beyond simple genetic calculation. Within this arrangement, the calf remains connected to the group through multiple pathways, not relying on a single individual, but supported by a network of relationships that operate simultaneously. Care is distributed, shared, and sustained across time, allowing the calf to develop within a system that balances absence and presence without interruption. As the mother returns from depth, the transition occurs seamlessly. The calf resumes contact, and the roles within the group adjust without visible coordination, maintaining a continuity that allows each individual to contribute to the survival of the whole. At a certain point in early life, the pattern that has held a young male within the group begins to loosen, not through any single visible event, but through a gradual separation that becomes permanent. Around 6 years of age, he leaves the family unit that shaped his first movements and interactions. Whether this departure is driven by subtle social cues or by an internal shift that aligns with age remains uncertain. What follows is a transition into a different kind of existence, one that unfolds outside the structure that continues without him. In the years that follow, young males do not move entirely alone. They gather in loose associations with others of similar age, forming temporary groupings that drift across open water. These bachelor groups are less cohesive than the tightly bonded family units, their composition changing over time as individuals move in and out. Gradually, their trajectories carry them toward colder regions, higher latitudes, where the conditions differ from the warmer waters they once inhabited. As they travel, their bodies continue to change. Growth does not plateau in the way it does for females. Over the next 2 decades, males increase in both length and mass, eventually reaching sizes that can exceed twice that of an adult female. Their heads expand significantly, developing the squared profile that distinguishes mature males. The spermaceti organ, already substantial, continues to enlarge throughout this period, contributing to the overall scale that defines their later form. Alongside these physical changes, their acoustic behavior shifts. Bachelor males are notably quieter than the family groups they left behind. The patterned sequences that define group communication become less frequent, and the structure of their cliques differs in measurable ways. The absence of dense social interaction appears to alter the way sound is used, with communication taking on a reduced or modified role in their daily activity. Time extends differently in this phase of life. For approximately 25 years, males remain largely separate from the social units of females and calves, moving across broad regions with limited interaction. Their movements cover great distances, guided by patterns that are still being mapped through intermittent tracking. Eventually, a shift occurs, and their paths begin to curve back toward the warmer waters where they were born. During breeding periods, mature males return to these regions, traveling distances that can exceed 5,000 miles. These journeys are not continuous migrations, but directed movements that bring them into contact with family groups for brief intervals. In earlier descriptions, such individuals were referred to as rogues, a term that reflected their solitary habits and infrequent appearances within social units. Their role during these encounters is narrowly defined. They mate, then depart. There is no integration into the group, no participation in the care of offspring, no sustained presence beyond the reproductive event. The structure of the family unit remains intact without them, continuing to function according to patterns established by the resident females. Despite the pronounced size difference between males and females, the mechanism by which mating occurs remains unclear. In many species, such dimorphism is associated with competition, displays, or direct conflict between males for access to females. In sperm whales, such behaviors have not been consistently observed. There are no clear records of sustained combat or elaborate display rituals within the context of mating. The selection process appears to operate through criteria that have not yet been fully identified. Within this arrangement, the male life path unfolds as a parallel system, separate from the continuity of the family unit, yet intersecting with it at specific moments. Growth, movement, and eventual return form a cycle that repeats across generations, shaped by patterns that remain only partially understood. On rare occasions, something drifts at the surface that does not resemble the usual fragments carried by currents. It moves slowly, shaped by time rather than by immediate formation, its texture uneven, its color ranging from pale gray to deep black. At first encounter, it offers little indication of what it is or where it came from, yet within it lies a connection to processes that unfold far below, hidden within the body of an animal rarely seen in full. This substance is known as ambergris. It originates inside the lower intestinal tract of the sperm whale, forming in a place where digestion meets resistance. The whale's diet includes large quantities of squid, and within those squid are hard, indigestible beaks that accumulate over time. Most of these pass through the digestive system without consequence, but in some cases, they become embedded within a growing mass, surrounded by layers of material that gradually solidify. As this accumulation develops, it takes on a waxy consistency, initially soft and irregular. The formation appears to be a response to the presence of these sharp, persistent fragments, though the exact biological mechanism remains unclear. It may serve as a protective process, isolating irritants within a cohesive structure that prevents damage to surrounding tissue. Alternatively, it may result from variations in digestion that lead to the gradual aggregation of material. Both interpretations align with observed properties, yet neither has been confirmed through direct study. Once formed, ambergris may remain within the whale for extended periods, increasing in size as additional layers accumulate. Over years, even decades, it can grow to substantial proportions. Pieces weighing more than 100 pounds have been recovered, suggesting long durations of development within a single individual. Eventually, the mass is expelled into the ocean, where it begins a transformation shaped by exposure rather than internal processes. Fresh ambergris carries a strong organic odor, reflecting its origin within the digestive system. As it floats or washes ashore, it is subjected to sunlight, salt water, and air, conditions that gradually alter its chemical composition. Over time, the scent changes, becoming more complex, less immediate, developing qualities that are described as sweet, earthy, and persistent. This transformation does not occur quickly. It unfolds over years as the material weathers and stabilizes. Historically, this altered form has been highly valued. Ambergris has been used in perfumery for centuries, not for its scent alone, but for its ability to fix and stabilize other fragrances, allowing them to last longer and evolve more gradually. One of the most well-known applications includes its use as a base component in formulations such as Chanel No. 5, where its properties contributed to the longevity and depth of the final composition. Despite its association with sperm whales, ambergris is not commonly found. It is estimated to occur in only about 1% of individuals, a rarity that contributes to its high value. The reasons behind this limited occurrence are not understood. There is no clear pattern linking its presence to age, diet, or environment, and no reliable method to predict which individuals will produce it. Because the process takes place entirely within the body and often over long periods, direct observation is not possible. Knowledge of ambergris comes from recovered samples and from examinations of stranded whales, where partially formed masses have occasionally been identified. These glimpses provide insight into its composition and growth, but they do not reveal the full sequence of events that leads to its formation. Within the broader context of the whale's life, ambergris remains an anomaly, a byproduct of digestion that transforms into something with significance beyond its origin. It drifts through the ocean as a fragment of an unseen process, shaped first by the internal conditions of the animal, then by the external forces of the environment, carrying within it a history that is only partially understood. There are moments when the familiar patterns of sound that define their presence begin to thin, not gradually across a group, but within a single individual whose activity has slowed beyond recovery. The surrounding water may still carry pulses from others nearby, yet from that one body, the signal fades until it is no longer detectable. What remains is not the absence of structure in the environment, but the absence of participation from the animal itself. This condition has been documented most clearly during stranding events, where individuals lie partially supported by shallow water, still alive yet unresponsive. Hydrophones placed close to these animals record almost complete acoustic silence. The mechanisms that normally produce clicks remain physically present, the tissues intact, the pathways unobstructed. Yet the output ceases, leaving a gap that cannot be explained by structural failure alone. Around the silent individual, the rest of the group behaves differently. Rather than reducing their own activity, nearby whales increase their vocal output, producing more frequent and more varied sequences. They circle the motionless body, maintaining proximity, their movements forming repeated loops that bring them close again and again. In some cases, they make physical contact, pressing against the still form, attempting to shift it, to lift it, to guide it upward. These efforts continue even when there is no visible response. The contrast between the silence of one and the activity of many creates a pattern that has been observed repeatedly yet remains unresolved. The cessation of sound does not align with visible damage to the structures responsible for producing it. In post-examination of stranded individuals, the anatomical components of the click system are often found to be intact. This suggests that the silence originates not from an inability to generate sound, but from a change in the processes that initiate it. One explanation considers the possibility of neurological shutdown. As the animal approaches the end of its life, the regions of the brain responsible for coordinating vocalization may cease to function, even while other systems remain active. This would place the silence within a sequence of internal changes where communication is among the first capabilities to decline. The absence of sound would then be a consequence of broader neurological failure, unfolding in stages that are not directly observable. Another interpretation approaches the silence from a different perspective. It considers that the absence of clicks may not be entirely involuntary, but instead reflects a behavioral shift that occurs at the threshold of death. In this view, the animal does not simply lose the ability to produce sound, but enters a state in which it no longer engages in the patterns that define its interactions with others. The silence becomes an action in itself, though one whose purpose remains unknown. No existing study has been able to distinguish clearly between these possibilities. The conditions under which these events occur limit the ability to observe internal processes in real time, and the available data is drawn from situations that are already atypical. Each recorded instance adds detail, but not resolution. Stranding events themselves often involve more than one individual. Groups arrive together or follow one another onto shore, forming clusters that reflect their social structure even in this altered environment. In 2016, 29 sperm whales were found stranded along the beaches of the North Sea within a single week. Most were young males, their bodies showing no immediate signs of injury or illness that would explain their presence on land. Their conditions suggested that whatever led them there operated at a level not easily detected through physical examination. Within these events, the silence of individual whales becomes part of a larger pattern that includes movement, proximity, and response. The absence of sound from one body does not halt the activity of others. Instead, it reshapes the interaction, creating a dynamic in which communication persists around a center that no longer participates. Along certain shorelines, the ocean delivers bodies that do not belong there. Animals built for depth resting in water too shallow to support their weight. The arrival is not always solitary. At times, multiple individuals appear within the same stretch of coast, their presence forming a pattern that repeats across regions and centuries without settling into a single explanation. Records of such events extend back at least to the 16th century. In 1577, Flemish artist Jan Wierix depicted a group of stranded whales along a European coastline, capturing an occurrence that had already drawn attention long before modern science began to document it systematically. Since then, similar events have been recorded across the world on nearly every continent where ocean meets land. The distribution is global, yet the underlying cause remains unresolved. Various explanations have been proposed, each addressing a different aspect of the phenomenon. One considers geomagnetic navigation. Sperm whales may rely in part on the Earth's magnetic field to orient themselves during long-distance movement. In certain coastal regions, magnetic field lines curve in ways that extend inland, creating a pathway that could lead an animal toward shallow water without a clear boundary to indicate the change. If navigation depends on this field, such distortions could result in gradual misdirection rather than sudden error. Another possibility focuses on sound. The ocean has become increasingly filled with artificial noise, including mid-frequency active sonar used in naval exercises. These signals travel long distances and can overlap with the frequencies used by marine mammals. In some documented cases, strandings have occurred in temporal proximity to such exercises, suggesting a connection between acoustic disturbance and disorientation. The Bahamas strandings of 2000, linked to a NATO exercise, provided one of the clearest associations, where multiple whales stranded shortly after exposure to sonar activity. Social behavior introduces an additional layer. Sperm whales maintain strong group cohesion, often responding collectively to changes in the condition of one individual. If a whale becomes disoriented or enters shallow water, others may follow, not through independent error, but through adherence to group structure. In this way, a single navigational mistake could propagate through an entire unit, resulting in multiple animals reaching the same unsuitable environment. There are also biological factors to consider. Infections affecting the inner ear could disrupt balance and spatial orientation, altering the animal's ability to interpret movement through water. Such conditions may not be externally visible, yet could influence navigation in ways that lead toward coastal areas. Evidence for this mechanism has been found in some stranded individuals, though it does not appear consistently across all cases. The events recorded in the North Sea in 2016 provide a detailed example. 29 sperm whales were found stranded within a single week, all identified as young males. Examination revealed that their stomachs contained significant amounts of plastic debris, including bags and other materials that had been ingested over time. In one individual, more than 30 plastic bags were present. While this indicates a clear interaction with human-produced waste, it does not fully explain the navigational failure. Other whales with minimal or no plastic ingestion have also stranded under similar circumstances. The presence of sonar as a contributing factor is supported in specific instances, yet strandings were documented long before such technologies existed. The depiction from 1577 confirms that the phenomenon predates modern acoustic interference by several centuries. No single explanation accounts for all occurrences, and each proposed mechanism addresses only part of the pattern. Across these events, the common element is not a single cause, but a convergence of possibilities, each influencing behavior under conditions that are difficult to reconstruct after the fact. The whales arrive at the shoreline through pathways that remain partially hidden, shaped by factors that interact in ways not yet fully understood. At times, the open water becomes a space of coordination rather than distance, where two highly capable species encounter one another under conditions that are not fully predictable. These encounters do not occur uniformly, nor do they follow a consistent pattern that can be easily mapped. Instead, they emerge in specific contexts, shaped by factors that remain only partially understood, leaving behind observations that suggest complexity without providing a complete explanation. Orcas are known to hunt sperm whales, though such events are relatively rare and often dependent on the composition of the group being targeted. When adult female sperm whales remain together in their established units, the outcome of these encounters tends to favor defense rather than predation. The group responds by forming a configuration known as a rosette, arranging themselves with their heads oriented inward and their tails extending outward. Calves are positioned at the center, enclosed within a ring of bodies that move in coordination. This formation is not improvised in the moment. It appears to be a learned behavior, one that can be executed by individuals with no prior direct experience of an attack. Calves that have not encountered orcas before are able to align themselves correctly when the group forms the structure, suggesting that the pattern is transmitted through observation and immediate response rather than through gradual trial. The speed with which this alignment occurs indicates a system of social learning that operates under conditions of heightened urgency. In contrast, solitary males, or loosely associated bachelor groups, do not possess the same level of coordinated defense. Without the structure provided by a cohesive unit, these individuals are more vulnerable when confronted by orcas. Encounters involving lone males are more likely to result in successful predation, as there is no collective formation to disrupt the approach of the attackers. A detailed observation recorded in 2016 off the coast of Sri Lanka provides a rare glimpse into the dynamics of such an event. A group of 35 orcas engaged with a cluster of 9 sperm whales over the course of approximately 4 hours. The interaction unfolded in phases, with the orcas maintaining pressure on the group, attempting to isolate a single individual. Eventually, they succeeded in separating a calf from the protective arrangement, leading to its capture. What followed did not align with the expectation of complete consumption. The orcas focused on specific parts of the carcass, particularly the tongue and the lower jaw, areas that contain high concentrations of energy. The remainder of the body was left largely untouched. This selective feeding pattern has been observed in other instances, indicating a preference that does not require the full utilization of the available biomass. Beyond the physical interaction, there is evidence that sperm whales distinguish between different orca populations based on their vocalizations. Recordings of orca calls have been played back to groups of sperm whales, producing varied responses depending on the origin of the sound. Calls from orca populations known to hunt sperm whales elicit changes in behavior, while calls from populations that primarily feed on fish do not produce the same reaction. This suggests that sperm whales are able to differentiate between types of orcas, recognizing patterns that correspond to varying levels of threat. The process by which this distinction is learned and maintained is not clearly defined. It may involve direct experience, where encounters shape future responses, or it may be transmitted socially within the group, allowing individuals to respond appropriately without prior exposure. The mechanism of this transmission, whether through acoustic learning, observation, or a combination of factors, remains an open area of study. Within these interactions, both species exhibit forms of coordination and recognition that extend beyond simple predator and prey dynamics. The encounters are shaped by learned behaviors, selective strategies, and forms of communication that influence the outcome in ways that are not yet fully understood. Before each pulse is released, a sequence unfolds inside the head that remains hidden from any external observation, a brief preparation measured in fractions of a second. The structures that will generate the sound align, the pathways clear, and at the same time another process occurs, one that prevents the whale from being affected by the very force it is about to produce. Without this internal coordination, each emission would carry consequences for the animal itself. At 230 decibels, the intensity of a sperm whale click exceeds levels that would damage most known auditory systems. Sound at that magnitude interacts not only with the environment, but with the body that creates it. In the absence of protective mechanisms, repeated exposure would degrade the structures responsible for hearing, altering perception in ways that would compromise survival. Yet, this does not occur. The whale continues to produce these signals throughout its life without apparent loss of auditory function. The explanation lies in a combination of anatomy and timing. The inner ear is not directly coupled to the skull in the same way it is in many terrestrial mammals. Instead, it is partially isolated by a layer of specialized tissue, a form of acoustic cushioning that reduces the transmission of vibration from the surrounding bone. This physical separation limits the amount of energy that reaches the sensory structures during each click. Alongside this structural feature, there is a neurological component that operates in precise synchrony with sound production. In the milliseconds immediately preceding each click, the auditory system undergoes a temporary suppression, reducing its sensitivity at the exact moment the signal is generated. This suppression is brief, reversible, and repeated with each pulse, creating a cycle in which perception is momentarily reduced, then restored as the echo returns. The sequence ensures that the outgoing signal does not interfere with the incoming information. The coordination required for this process is exact. The timing must align with the production of each click, and the transition between suppression and sensitivity must occur without delay. The result is a system in which the whale can emit a powerful signal while remaining capable of detecting the subtle variations in the echoes that follow. The process resembles a mechanism that anticipates its own effect, adjusting internal conditions in advance of external action. Evidence suggests that the protective aspects of this system may have developed before the full capacity of the click mechanism itself. The anatomical isolation of the inner ear appears to precede the evolution of the high-intensity signals, indicating that the sensory system adapted in a way that allowed for later amplification. In this sense, the capacity to generate powerful sound may have emerged within a framework that was already prepared to accommodate it. Within the same environment, another lineage has been evolving under the influence of these interactions. Giant squid, occupying the depths where these encounters occur, possess eyes of exceptional size and sensitivity. These organs are capable of detecting faint changes in the surrounding water, including disturbances caused by movement. As a whale approaches, it alters the local field of bioluminescent particles, creating patterns that can be perceived before direct contact. This relationship suggests a gradual adjustment on both sides. As the whale's ability to produce focused, high-energy clicks becomes more refined, the squid's capacity to detect approaching motion becomes more sensitive. Each adaptation influences the other, shaping a dynamic that unfolds over long periods without direct observation. The darkness of their shared environment does not prevent interaction, but it conceals the process by which these changes accumulate. There is no continuous record of this development preserved in physical form. Squid, lacking hard skeletal structures, leave little trace in the fossil record. The history of their interaction with sperm whales must be inferred from the anatomy that exists today, from the alignment of features that suggest a shared influence. Within these forms, the evidence of an ongoing adjustment remains, a quiet progression shaped by contact that occurs beyond the reach of direct observation. Time leaves marks that do not fade with movement, accumulating quietly within structures that continue to function while carrying a record of what has already passed. In sperm whales, these traces are not stored in a single place, but distributed across different parts of the body, each preserving a fragment of experience in a form that can later be interpreted. What appears as a living organism is also, in many ways, an archive assembled over decades. Within the teeth, this record is layered with a regularity that reflects the passage of years. Growth layer groups form as successive deposits of dentine, building one upon another in a sequence that can be counted to estimate age. Each layer corresponds to a period of time, creating a structure that extends from early development through maturity. When examined closely, these layers reveal more than duration. They contain variations in chemical composition that correspond to changes in diet and environment. Through stable isotope analysis, researchers can extract information from these layers that reflects where the whale has been and what it has consumed. Different regions of the ocean carry distinct isotopic signatures, shaped by variations in water chemistry and biological activity. As the whale feeds within these regions, those signatures become incorporated into the dentine, preserved in a form that remains stable over time. By reading these patterns, it becomes possible to reconstruct aspects of the whale's movements across years that were never directly observed. The surface of the body carries a different kind of record, one that reflects direct interaction with other organisms. Scars from encounters with large squid appear as circular impressions, their size and arrangement corresponding to the suction cups of the animal that produced them. Some of these marks exceed 4 inches in diameter, indicating contact with species that inhabit depths beyond 3,000 feet. Each scar represents a moment of contact that cannot be witnessed, yet remains visible long after the event has passed. Beneath the skin, additional information is stored within the blubber. This layer, often sampled through small biopsy procedures, contains chemical indicators that reflect physiological states over time. Hormones embedded within the tissue can reveal patterns of stress, with elevated cortisol levels indicating prolonged exposure to challenging conditions. Other compounds provide evidence of reproductive history, marking periods of pregnancy and recovery. These signals do not capture specific events, but they trace broader patterns that unfold across the life of the animal. More recently, another element has become part of this internal record. Microplastics, dispersed throughout the global ocean, have been detected within the blubber of sperm whales from every major basin. Samples collected from remote regions, including the Central Pacific, far from any direct source of pollution, contain these particles embedded within the tissue. Their presence reflects a change in the environment that has become widespread enough to reach even the most distant populations. Each of these elements contributes to a composite record that exists within the body itself. Age, movement, interaction, physiological condition, and environmental exposure are all preserved in different forms, accessible through methods that continue to develop. Yet much of this information can only be fully examined after the animal's life has ended. Post-mortem analysis provides the most complete view, allowing structures to be studied in detail that cannot be accessed in a living individual. In this way, the whale carries within it a layered history, one that is present throughout its life, but only partially readable until it has passed. The body retains these traces without interruption, forming a record that grows with each year, accumulating details that extend beyond direct observation. Knowledge of this animal does not accumulate in a continuous stream, but in fragments gathered under conditions that rarely align with the full scope of its life. The difficulty is not a lack of interest or effort, but a matter of location and duration. Sperm whales spend most of their time far below the surface, in regions where light does not reach, and they do so far from land where observation can be sustained. These three conditions, depth, distance, and submergence, create a setting where direct study becomes intermittent by necessity. When a whale surfaces, it offers a brief window of accessibility, and within that window, researchers attempt to gather information that can extend beyond the moment. One method involves attaching tracking devices to the animal's body. These tags record movement, depth, and sometimes sound, transmitting or storing data that can be analyzed later. The attachment is temporary. Tags are designed to detach after a period of time to prevent long-term interference with the animal's behavior. As a result, each dataset is limited in duration, often lasting weeks before the device separates and is either recovered or lost. Another approach focuses on collecting small samples of tissue without causing significant harm. Biopsy darts are used for this purpose, launched from crossbows toward whales at the surface. Each successful sample requires a sequence of precise conditions. The animal must be located in open water, approached carefully to avoid triggering avoidance behavior, and sampled accurately from a moving platform. The fragment obtained, usually skin and blubber, provides material for genetic analysis, chemical study, and assessment of physiological state. The process yields valuable data, but it represents only a small portion of the information contained within the animal. To understand behavior that occurs beyond the surface, acoustic recording becomes essential. Hydrophone arrays are deployed across sections of the ocean, capturing the sounds produced by whales as they move through the water. These systems allow researchers to monitor presence, communication patterns, and movement over larger areas. The deployment of such arrays involves considerable technical and logistical effort, and the recordings are often affected by other sources of noise, including shipping activity and environmental factors that alter sound transmission. For deeper insight into submerged activity, data loggers are attached directly to whales. These devices record parameters such as depth, orientation, and acoustic output, providing a continuous record of movement through the water column. However, they do not capture visual information. The internal perspective of the whale, the exact nature of its interactions at depth, remains beyond the reach of these instruments. The data describes patterns, but not the full context in which those patterns occur. Despite centuries of observation, certain fundamental events have never been directly recorded. No documented instance exists of a sperm whale giving birth in its natural environment. The process has been inferred from indirect evidence and from observations of calves at various stages of development, but the event itself remains unseen. Similarly, natural death within the open ocean has not been observed. All recorded deaths involve circumstances that bring the animal to the surface or to shore, such as strandings, collisions with vessels, or entanglement in fishing gear. Other central aspects of life follow the same pattern. Deep hunting behavior is understood through traces and data, rather than direct visual confirmation. Mating occurs without observation, inferred from population dynamics and genetic relationships rather than from witnessed interaction. Each of these events is essential to the continuity of the species, yet each remains outside the scope of sustained study. What emerges from these limitations is a form of understanding built from partial views, where each method contributes a segment of information that must be integrated with others. The absence of direct observation does not prevent knowledge from forming, but it shapes the way that knowledge develops, requiring interpretation, inference, and the careful alignment of data gathered under conditions that reveal only portions of a larger, continuous life. In recent years, A different kind of listening has taken shape, one that extends beyond recording sound and begins to search for structure within it. The signals that once appeared as repeating patterns are now being approached as a possible system, not yet understood, but potentially consistent enough to be analyzed at scale. This effort does not rely on a single discipline, but draws together methods from fields that do not usually intersect. Project SETI, the Cetacean Translation Initiative, began in 2020 with a defined objective to examine whether the acoustic patterns produced by sperm whales can be interpreted through computational analysis. The project brings together marine biologists, linguists, cryptographers, and researchers in artificial intelligence, forming a collaboration shaped by the recognition that traditional biological approaches alone may not be sufficient to address the problem. Each field contributes a different perspective, from the study of animal behavior to the analysis of complex signal systems. The work centers on a population of sperm whales located in the waters off Dominica in the Eastern Caribbean. This group, consisting of approximately 40 individuals, has been observed continuously since 2005 under the research of Shane Garrow. Over time, each whale has been identified through a combination of physical markings, acoustic patterns, and social relationships, creating one of the most detailed records of a sperm whale community currently available. The continuity of observation allows for a level of contextual understanding that is rare in open ocean research. To capture the data needed for analysis, arrays of underwater hydrophones are deployed across the region. These instruments record the clicks and codas produced by the whales, creating a continuous stream of acoustic information. The recordings are not treated as isolated sounds, but are paired with observations of behavior, identifying which individuals are present, which are producing the sounds, and what interactions are occurring at the time. This pairing of signal and context forms the basis of a dataset designed for computational interpretation. The scale of this dataset is central to the project. The aim is to collect on the order of 4 billion coda clicks, a volume large enough to allow statistical patterns to emerge through machine learning techniques. By training models on this data, researchers hope to identify recurring structures that may correspond to specific contexts or interactions. If such correspondences exist, they could indicate that the codas carry information beyond simple repetition. The underlying assumption is that meaning, if present, may be detectable through patterns that align consistently with observable conditions. A sequence of clicks that appears regularly in association with a particular behavior could suggest a form of reference, a connection between sound and context. Over time, these associations might reveal elements of structure analogous to features found in human language, such as syntax or discrete units of meaning. At the same time, there is recognition that this assumption may not hold. The structure of sperm whale communication may not conform to the frameworks used to analyze human language. The signals may operate on principles that differ fundamentally, lacking clear boundaries between units or relationships that map onto familiar linguistic categories. In such a case, the analytical tools being applied may need to be adjusted or redefined to match the nature of the system being studied. As of early 2025, the process remains ongoing. Data continues to be collected, models continue to be trained, and patterns are examined without a confirmed translation. The effort proceeds within a space where the presence of structure is evident, yet its interpretation remains incomplete, shaped by methods that are still adapting to the signals they are designed to analyze. Even if patterns begin to resolve into structure and correlations settle into something that resembles consistency, the question of what that structure represents remains suspended at a different level. A sequence can be identified, repeated, and associated with a particular context, yet the step from pattern to meaning does not follow automatically. It requires an assumption, one that is often left implicit, that whatever is being conveyed can be recognized within the framework used to interpret it. The effort to decode sperm whale communication carries within it this assumption that meaning, if it exists, will take a form that can be translated into human terms. The models being trained are designed to detect relationships between signals and events, to identify recurring associations that suggest reference or intention. These methods rely on parallels with known systems, particularly human language, where structure can be mapped onto meaning through shared categories and definitions. Yet there is no guarantee that such parallels apply in this case. The difficulty lies not only in the complexity of the signals, but in the nature of perception itself. Philosopher Thomas Nagel, in his 1974 paper on the subjective experience of bats, proposed that the internal world of an echolocating animal may be inaccessible not because of insufficient data, but because of a difference in the structure of experience. An organism that constructs its environment primarily through reflected sound does not simply receive different information, it organizes that information in ways that may not align with human cognition. A sperm whale moves through a space defined by acoustic reflection, forming a continuous model of its surroundings through returning signals. Depth, distance, and texture are integrated into a perception that unfolds without reliance on light. Social interaction is layered within this same system, encoded in sequences of clicks that carry information between individuals. The sensory and communicative processes are intertwined, creating an internal representation that may not separate environment from interaction in the same way human perception does. Linguistic theory introduces a further consideration. Benjamin Lee Whorf proposed that the structure of language influences the way its speakers categorize and understand the world. If the codas used by sperm whales encode distinctions that do not exist in human language, then any attempt at translation may encounter concepts that have no direct equivalent. The output of such a translation might appear incomplete or unfamiliar, not because it lacks meaning, but because the categories it expresses do not align with those used in human thought. Within this context, the process of decoding becomes more than a technical challenge. It becomes a question of whether interpretation is possible within the limits of the frameworks being applied. The patterns identified through machine learning may reveal regularities, relationships, and structures that correspond to specific conditions or interactions. These findings can demonstrate that information is being conveyed, that the signals are not random, and that they operate within a consistent system. However, identifying that information exists does not necessarily provide access to its content. A pattern may correlate with a particular behavior, indicating that it plays a role in coordination or response, yet the internal significance of that pattern to the whale may remain undefined. The signal can be mapped, its occurrence predicted, its association described, while its experiential meaning remains outside direct reach. This does not diminish the value of the analysis. It clarifies the boundary within which interpretation operates. The project may succeed in demonstrating that codas carry structured information and in describing the rules that govern their use. At the same time, the question of what that information represents within the whale's own perception may persist, shaped by differences that are not easily translated across species. The water that carries their cliques has begun to change in ways that cannot be sensed all at once, only accumulated over time. Subtle shifts layered year after year into the medium they depend on. For most of their evolutionary history, the chemistry of the ocean moved slowly, buffered by geological cycles that unfolded over millions of years. In the span of a few human generations, that balance has begun to tilt, altering conditions at a pace that has no clear parallel in the recent fossil record. One of the quietest shifts lies in the chemistry itself. Since industrialization, the average ocean pH has fallen from 8.2 to approximately 8.1, a change that corresponds to a roughly 26% increase in acidity. The difference appears small in notation, yet it alters the behavior of dissolved carbon and the structure of calcium-based tissues. Many squid species, which form the primary diet of sperm whales, rely on delicate biological processes that are sensitive to these changes. Their development, buoyancy, and distribution can be affected in ways that are still being measured, each adjustment rippling upward through the food web. Temperature adds another layer to this transformation. As surface waters warm, the structure of the ocean reorganizes vertically. One consequence is the expansion and deepening of the oxygen minimum zone, a layer where dissolved oxygen levels fall low enough to shape the distribution of marine life. Many deep-sea organisms cluster around the boundaries of this zone, where conditions are balanced between scarcity and survival. As the zone shifts, these boundaries move with it, carrying prey populations into new depths and regions. The routes and feeding grounds that have been followed for decades may no longer align with these shifting patterns. Sound, which forms the primary channel through which sperm whales perceive and communicate, is also changing. Since the middle of the 20th century, ambient ocean noise has increased steadily, with estimates suggesting a doubling in some regions each decade. The sources are varied, from global shipping networks to naval sonar, seismic exploration, and offshore construction. These sounds travel efficiently through water, filling spaces that were once comparatively quiet. In areas of intense activity, sperm whales have been recorded altering the structure of their Kodas, adjusting timing and spacing in ways that suggest an attempt to remain detectable within a noisier environment. Alongside these changes, material carried into the ocean accumulates within the bodies of its inhabitants. Microplastics, reduced to fragments small enough to move through water columns and biological tissues, have been found in individuals sampled across every major ocean basin. Larger debris appears in more visible forms, recovered from the stomachs of stranded animals in quantities that indicate repeated ingestion over time. These materials do not belong to the cycles that shaped marine life, and their long-term effects remain only partially understood. Each of these pressures unfolds on its own scale: chemical, thermal, acoustic, physical, yet they converge within a single organism that lives for decades and depends on learned knowledge passed across generations. A matriarch's memory reflects an ocean that may no longer behave as it once did, while younger individuals inherit patterns that must adapt in real time. The system that connects environment, perception, and culture continues to function, yet the conditions it operates within are shifting in ways that are difficult to isolate or predict. It began not as an industry, but as an encounter shaped by weather and chance. A vessel pushed beyond familiar routes into waters where large, dark forms moved just beneath the surface. In 1712, a Nantucket ship, driven offshore by a storm, found itself among sperm whales in numbers that had not been recorded in such proximity before. What followed was not immediate transformation, but recognition. The animals carried a resource that could be rendered, stored, traded, and scaled. From that moment, pursuit became organized, and over the following decades, the ocean itself became a mapped field of extraction. By the turn of the 19th century, American whaling fleets had extended far into the Pacific, tracing migratory paths that had once existed beyond the reach of sustained human presence. Voyages stretched across years, ships operating as floating factories, processing oil on open decks beneath shifting skies. The world described in Herman Melville's Moby Dick, published in 1851, reflects a system already approaching its fullest extent, a network of routes, techniques, and expectations refined through repetition. Sperm whales became central to this system, their spermaceti oil valued for its clarity and stability, their bodies reduced to components measured in barrels and yield. Across the span from 1800 to 1987, when the International Whaling Commission moratorium came fully into effect, an estimated 800,000 sperm whales were killed. The number moves easily across the page, but its weight resides in what it represents over time. Each individual carried not only its own physiology, but a position within a structure of relationships, a continuity of learned behavior that extended backward through generations. Removal occurred unevenly, often targeting the largest animals, those that surfaced predictably, those that led. Within family groups, the loss of older females altered patterns that had been maintained through repeated transmission. A matriarch's role is not written in a single act, but in accumulated knowledge, the memory of routes, feeding grounds, seasonal shifts, responses to threats that may arise only once in decades. When such an individual is removed, there is no external record to replace what is gone. The knowledge persists only if it has already been distributed, and even then, it becomes partial, less precise, subject to gradual erosion. Acoustic traditions reflect this change in ways that can be measured. Comparisons between historical recordings and contemporary observations suggest that modern sperm whale groups exhibit reduced Koda diversity. Patterns that were once distinct to particular families or regions appear to have diminished or disappeared entirely. These were not random variations, but structured sequences, learned and shared, forming part of a system through which identity and coordination were maintained. Their absence is not silence, but simplification, a narrowing of expression shaped by interruption. The scale of removal also affected population structure more broadly. Social networks that once extended across large areas became fragmented, densities reduced, encounters less frequent. Recovery, where it occurs, proceeds within these altered conditions, new generations forming connections without access to the full range of inherited behaviors that once existed. The ocean remains vast, but the distribution of experience within it has shifted. What was taken during these centuries cannot be fully described through numbers alone. The loss includes individuals, certainly, but also the accumulated outcomes of their lives, the decisions repeated across time, the adjustments refined through observation and memory. These were not stored in a central place, not written or recorded in a way that could be retrieved. They existed only in living systems, carried forward through behavior, through association, through sound. When those systems were broken, the continuity they maintained did not pause. It ended, and what followed began from a diminished foundation, moving forward without access to what had been known before. The end of large-scale hunting did not arrive as a sudden quiet, but as a gradual easing, a reduction in pressure that unfolded unevenly across oceans already altered by centuries of pursuit. In 1986, the International Whaling Commission moratorium marked a turning point, though not an absolute one. Some nations continued whaling under exemptions, yet the coordinated industrial effort that had once spanned hemispheres began to recede. What remained was a population no longer being removed at the same rate, set within an ocean that had already absorbed the absence of so many individuals. Recovery, in this context, does not resemble a rapid return, but a slow recalibration shaped by the biology of the animal itself. Sperm whales reproduce on a timescale that resists acceleration. Females reach sexual maturity at around 9 years of age, and after a gestation period lasting between 14 and 16 months, a single calf is born. That calf remains dependent for extended periods, and the interval between births typically spans 4 to 6 years. These rhythms are not easily compressed, and they define the pace at which numbers can increase, even under ideal conditions. Observed growth rates in recovering cetacean populations rarely exceed 7% per year, and for sperm whales, the figure is likely lower. When applied across a global population that had been reduced over generations, the implications extend far beyond a single human lifetime. Even in the absence of further losses, projections suggest that a full return to pre-whaling abundance would require between 200 and 400 years. This is not a delay caused by inaction, but a reflection of the species' own life history, its reliance on longevity, extended care, and stable social structures. The pattern of recovery is not evenly distributed. In some regions, particularly within parts of the North Atlantic, there are indications of gradual increase, groups becoming more frequently encountered, numbers stabilizing and slowly rising. In other areas, populations remain at levels that reflect historical depletion, with fewer individuals spread across broader ranges. These differences are shaped by local conditions, by the degree of past exploitation, and by the ongoing pressures that continue to influence survival and reproduction. Alongside the return of numbers, another process unfolds more quietly, one that is less visible but equally significant. The restoration of population size does not immediately restore the structures that once existed within it. Cultural transmission, the passing of knowledge through generations, depends on continuity, on the presence of individuals who carry and share accumulated experience. When that continuity is broken, it does not reform automatically as numbers increase. A population may reach a point of numerical stability while still lacking the full range of behaviors that once defined it. Acoustic traditions, patterns of movement, specialized foraging techniques, all require time to re-emerge, if they re-emerge at all. Some may persist in fragments, held within smaller groups, gradually spreading as interactions increase. Others may have been lost entirely, leaving spaces that new behaviors may eventually fill, though not in identical form. The first generations born into this recovering landscape inherit a different context from those that came before. They are shaped by a world where elders are fewer, where the density of knowledge is reduced, where pathways once reinforced through repetition may no longer be as clearly defined. Their learning still occurs within family groups, still guided by association and observation, yet the range of what can be transmitted has shifted. Over time, these generations become the carriers of what remains and what is newly formed. The process is continuous, extending forward without a clear boundary between loss and renewal. What returns is not a replication of what was, but a living system adapting within altered conditions, rebuilding structure at a pace set not by urgency, but by the steady progression of life itself. There is a movement that repeats across their lives, a slow vertical exchange between depths where light does not reach, and surfaces where the atmosphere meets the sea. It is not a single journey, but a cycle, carried out thousands of times over decades, each descent and return linking layers of the ocean that would otherwise remain more isolated. In this motion, something less visible is carried upward, not as an intention, but as a consequence of feeding and survival. At depth, sperm whales consume prey that draw their energy from ecosystems structured around scarcity, where nutrients accumulate differently than they do near the surface. When the whale rises to breathe, it also releases waste into waters where those same nutrients are limited. The material is rich in iron and nitrogen, elements that play a critical role in biological productivity but are often in short supply in sunlit regions. This transfer, repeated across individuals and across time, forms what researchers have come to describe as a biological pump, driven not by currents or tides, but by the bodies of the animals themselves. At the surface, these nutrients become available to phytoplankton, microscopic organisms that form the base of the marine food web. Their growth depends on sunlight, carbon dioxide, and the availability of trace elements like iron. When conditions align, phytoplankton populations expand, drawing carbon dioxide from the atmosphere through photosynthesis and releasing oxygen as a byproduct. The scale of this process extends beyond immediate surroundings, contributing to broader cycles that influence atmospheric composition. A study conducted in 2010 focused on the Southern Ocean estimated that sperm whales, through this process of nutrient redistribution, stimulate phytoplankton growth sufficient to sequester approximately 400,000 tons of carbon per year. This figure exceeds the carbon output associated with the whale's own respiration, indicating that, over time, their presence contributes to a net reduction of carbon dioxide in the atmosphere. The effect emerges not from a single action, but from the accumulation of countless cycles, each one modest, yet collectively significant. Before the era of industrial whaling, global sperm whale populations were estimated at around 1.1 million individuals. Within that larger population, the same processes were occurring continuously, distributed across oceans, integrated into the natural rhythms of marine systems. The removal of approximately 800,000 whales did not only reduce numbers within a species, it altered the scale at which this nutrient transport occurred. Fewer individuals meant fewer vertical exchanges, less redistribution of iron and nitrogen, and a diminished capacity to support phytoplankton growth in certain regions. The implications extend beyond immediate ecological relationships. Phytoplankton play a central role in regulating atmospheric carbon, and changes in their abundance can influence the balance between carbon uptake and release. The reduction of whale-driven nutrient cycling represents a shift within this larger system, one that was not recognized at the time the removals were taking place. Only in recent decades has this connection been examined in detail, revealing an interaction between animal behavior and global processes that had previously been considered separately. The presence of sperm whales therefore contributes to the functioning of the ocean in ways that are not immediately visible. Their movements connect layers, their feeding links distant ecosystems, and their biology participates in cycles that extend from the depths to the atmosphere. When their numbers decline, the effects move outward, altering patterns that depend on their continued activity. What emerges is a view of the ocean not as a collection of isolated parts, but as a system shaped by interactions that cross boundaries of depth and scale. Within that system, the role of a single species can influence processes that reach far beyond its immediate environment, creating connections that only become apparent when they are disrupted. Long before hydrophones were lowered into the water or data loggers attached to moving bodies, the image of this animal had already been shaped in language, carried not through measurement but through narrative. In 1851, Herman Melville published Moby Dick, drawing on 18 months of lived experience aboard whaling vessels. Within its pages are observations that align closely with what later science would confirm, details of behavior, anatomy, and encounter that remain recognizable even now. Alongside these are elements that belong not to observation, but to interpretation, a layering of symbolism and invention that transformed the whale into something larger than an organism. The figure that emerged from this blending was not neutral. It was vast, elusive, and often framed as adversarial, an embodiment of resistance within a landscape already defined by pursuit. For more than a century, this portrayal shaped how the animal was understood beyond the boundaries of literature. The sperm whale entered Western imagination not as a participant in an ecosystem, but as a presence that opposed, challenged, or evaded human intent. The distinction between observation and narrative blurred, and in that space, certain lines of inquiry remained unexplored. Scientific investigation does not occur in isolation from the culture that surrounds it. The questions that are considered worth asking are influenced by the assumptions carried into the field, often without explicit recognition. For much of the 20th century, research on large whales focused on distribution, migration, and population dynamics, areas that aligned with management and resource assessment. The possibility that these animals possessed complex internal states or structured communication systems did not occupy the center of inquiry. It existed at the margins, occasionally noted, rarely pursued in depth. A shift began in 1971 when Roger Payne documented the songs of humpback whales and presented them not only within scientific literature, but also as recordings accessible to the public. The sounds themselves carried a structure that was immediately apparent, sequences that unfolded with variation and repetition, patterns that suggested organization beyond simple signaling. The response extended beyond academic circles, altering public perception and, in turn, influencing the direction of research. Whales became associated with communication, with continuity, with qualities that invited closer attention. Within this changing framework, not all species were received in the same way. The vocalizations of humpbacks, with their tonal qualities and extended sequences, were more readily interpreted as expressive. Sperm whale clicks, by contrast, present as brief, percussive signals, rapid and dense, lacking the extended melodic contours that had captured attention elsewhere. The difference in acoustic character shaped perception, guiding interest toward what seemed more immediately accessible. Research priorities followed these contours. Over the following decades, humpback whale song became one of the most extensively studied forms of animal communication, examined across populations and generations. Sperm whale codas, though recognized as structured, remained less explored, in part due to the technical challenges involved, and in part due to the absence of a framework that readily accommodated their interpretation. The questions asked of one species were not applied with equal intensity to another, even when the underlying complexity may have been comparable. The influence of narrative persists in subtle ways. The stories that define an animal's place within human thought can guide attention, shaping which signals are amplified and which remain peripheral. As research tools expand and datasets grow, these influences become more visible, revealing the extent to which earlier assumptions directed the path of inquiry. What is now emerging is not only new information about the animals themselves, but also a reflection of how understanding has been filtered through inherited perspectives, gradually shifting as those perspectives are reconsidered. Measured against the span of their existence, the period in which humans have attempted to understand the ocean occupies only a narrow margin, a brief interval set against tens of millions of years of continuous presence. Sperm whales have moved through shifting basins and changing climates, following patterns that have adjusted gradually across epochs. Modern oceanography, by contrast, extends back roughly 150 years, its instruments and models refining a picture that remains partial, constructed from fragments gathered at the surface and from short encounters below. Within that disparity of timescales lies a difference in perception. The whale does not rely on light to define its surroundings, but on sound, emitted and received in rapid succession, forming a three-dimensional representation built from echoes returning through water. Each click carries information outward. Each reflection returns with subtle variations that encode distance, density, movement. This process unfolds continuously, allowing the animal to navigate spaces that remain inaccessible to direct human observation, not as a map drawn once, but as a dynamic field that updates moment by moment. Alongside this acoustic system, there are indications of additional sensory capacities that operate beyond the limits of human perception. Magnetite crystals have been identified within cetacean tissue, suggesting a mechanism through which the Earth's magnetic field may be detected. If functional, such a system would provide a form of orientation that does not depend on external markers, a reference that persists regardless of visibility or depth. The extent to which this sense contributes to navigation remains uncertain, yet its presence points to layers of perception that are not directly accessible through human experience. The structure of the brain adds another dimension to this difference. Sperm whales possess brains that can weigh up to 20 pounds, with a well-developed limbic system associated with emotional processing. In addition, they contain a paralimbic lobe, a region not found in terrestrial mammals, including humans. Its function has not been determined, and its role within cognition remains an open area of study. The existence of such structures suggests capacities that have evolved along a distinct path, shaped by environments and pressures that differ from those encountered on land. Across evolutionary time, these biological systems have been carried through periods of significant planetary change. The lineage that leads to modern sperm whales extends back approximately 30 million years, encompassing intervals of glaciation, shifts in sea level, and reconfiguration of ocean currents. The species that exists today has persisted through these transitions, maintaining continuity while adapting to altered conditions. The accumulation of this history is not stored in a single location, but distributed across physiology, behavior, and social transmission. If knowledge can be understood as the integration of repeated experience, then a species with such duration may carry forms of understanding embedded within its structure. These would not resemble records in the human sense, not written or stored in discrete units, but expressed through tendencies, preferences, responses that have been refined over long periods. Cultural transmission within family groups adds another layer, allowing information to persist beyond individual lifespans, carried forward through association and learning. Access to this accumulated experience presents a challenge. The systems through which it is expressed do not align directly with human frameworks of interpretation. Acoustic signals, neural structures, and behavioral patterns offer points of contact, yet they do not translate easily into categories that can be fully understood from the outside. The possibility remains that aspects of the ocean, as encountered by these animals, exist in forms that cannot be directly mapped onto human perception. What can be observed is the presence of structure, the integration of sensory systems, neural capacity, and social continuity operating across time. Within that structure lies the potential for forms of knowledge shaped by conditions that have persisted far longer than human study, embedded in ways that are only beginning to be approached through careful observation and evolving methods. Within the layered folds of their brain lies a region that does not align with familiar maps, a structure identified not through behavior but through careful comparison of anatomy across species. In 2006, neurobiologist Patrick Hoff and colleague Estelle van der Gucht described the presence of spindle-shaped neurons, known as von Economo neurons, in several cetacean species, including sperm whales. These cells are relatively rare across the animal kingdom, yet they appear consistently in species whose lives depend on complex social interaction. In humans, von Economo neurons are concentrated within the anterior cingulate cortex and the frontal insula, areas associated with rapid, intuitive judgment, social awareness, and the integration of emotional information. They are also present in great apes, elephants, and certain bird lineages, each group characterized by intricate social structures and the need to respond quickly to the behavior of others. Their appearance in cetaceans suggests that similar pressures, though separated by millions of years of evolutionary distance, have led to comparable neural solutions, a process often described as convergent evolution. The implication is not that these species think in the same way, but that their nervous systems have developed mechanisms capable of supporting high levels of social coordination. For animals that live in stable groups, where relationships persist over decades and communication must operate across distance and time, such mechanisms would be advantageous. The presence of these neurons indicates a capacity for processing information that extends beyond simple stimulus and response, allowing for patterns of interaction that depend on memory, recognition, and rapid assessment. Alongside these shared features, however, cetacean brains contain structures that do not have clear equivalents in terrestrial mammals. One such region is the paralimbic lobe, a distinct cortical area that has been identified through neuroanatomical study but remains without a defined function. It occupies a position that connects sensory input with regions associated with emotional processing, suggesting a role in integrating different streams of information, yet its specific operations have not been determined. In sperm whales, this paralimbic lobe is proportionally larger than in any other cetacean examined. Its scale alone indicates that it is not a minor addition, but a substantial component of the brain's architecture. The pathways that connect it to other regions imply that it participates in processes that are central rather than peripheral, receiving signals that reflect both external conditions and internal states. What emerges from this integration is not directly observable, as the outputs of this region cannot be isolated in a way that corresponds to human categories. One possibility is that it contributes to the processing of acoustic information, particularly the structured sequences of clicks that define social interaction within groups. These signals carry timing, spacing, and patterning that may encode layers of meaning not easily separated into discrete units. A neural system specialized for handling such information could operate in ways that differ from the processing of spoken language, organizing perception around patterns that unfold over very short timescales, yet remain consistent across repeated exchanges. Another possibility is that the paralimbic lobe supports forms of internal experience that do not map onto human analogs. Emotional states, social awareness, and perception may be integrated in configurations that reflect the demands of life in a three-dimensional, acoustically mediated environment. Without direct access to these processes, interpretation remains limited to inference, guided by structure rather than direct observation. The presence of this region serves as a reminder that the architecture of cognition is not uniform across species. While certain elements may converge, others diverge, shaped by environments and evolutionary histories that differ fundamentally. In the case of the sperm whale, the paralimbic lobe stands as a feature that signals both complexity and uncertainty, an indication that there are aspects of their inner organization that extend beyond current understanding, embedded within a system that continues to operate without revealing its full nature. There is a boundary that does not announce itself with a line, but with a quiet accumulation of things that remain unresolved, observations that fit together structurally yet refuse to settle into explanation. Sperm whales occupy that boundary, not as anomalies, but as organisms whose coherence is clear even as their interpretation remains incomplete. Their bodies, their signals, their societies all present patterns that can be measured, recorded, and compared, yet the integration of those patterns into a single, stable understanding remains out of reach. Much of modern science was constructed to study systems that do not possess interior experience in any meaningful sense, or whose internal states can be reduced to measurable outputs without significant loss of meaning. Physics describes motion, energy, and interaction without needing to account for perception. Chemistry explains transformation at the level of molecules, indifferent to awareness. Even large-scale biological processes, when viewed through this lens, can be organized into cycles, feedback systems, and adaptive responses that remain external to the organism itself. As inquiry moves toward animals whose lives depend on sustained social interaction, memory, and communication, the limitations of that framework begin to surface. The question is no longer limited to what structures do, but extends toward what they correspond to internally. Behavior can be recorded, neural activity can be mapped, acoustic signals can be cataloged, yet the translation between these domains and any form of lived experience is not direct. The gap is not one of missing data alone, but of conceptual reach. Across the history of scientific thought, similar gaps have appeared and gradually narrowed. The position of the Earth was recontextualized within a larger system, not by abandoning observation, but by extending it beyond immediate perception. The origins of species were reframed through evolutionary processes that operated over timescales far longer than human intuition could easily grasp. The brain itself, once treated as a uniform organ, revealed layers of specialization and complexity that required new methods and new language to describe. Sperm whales now sit within a similar interval, where the available evidence suggests a level of organization that exceeds current explanatory tools. Their acoustic behavior carries structure that can be quantified but not yet interpreted. Their neural architecture contains regions that can be described anatomically but not functionally. Their social systems exhibit continuity across generations, yet the mechanisms of transmission remain only partially understood. Each of these elements is stable in itself, yet their convergence produces a system that resists reduction. The presence of such resistance does not indicate failure, but direction. It marks the point at which existing methods begin to lose resolution, where new approaches become necessary not because the subject is inaccessible, but because the framework applied to it is incomplete. Every unresolved observation defines a path outward, suggesting where attention might shift, where tools might evolve, where concepts might expand to accommodate forms of organization not previously considered. In 2024, researchers working with long-term acoustic datasets recorded an interaction that did not align with established patterns. A sperm whale, exposed to a playback of its own Kodas sequence, produced a response that diverged from known repertoires, a variation that had not been documented in prior recordings. The exchange did not fit cleanly into categories of repetition or mimicry. It suggested a form of response that incorporated the original pattern while extending it into something new. The data remain under analysis, the interpretation unresolved, the significance uncertain. Yet the event exists within the record, another point along the boundary where description is possible but understanding has not yet settled. It joins the many other fragments that define this edge, each one precise in itself, each one incomplete in relation to the whole, forming a continuity that extends beyond the limits of current explanation while remaining fully grounded in the observable world. Long before coastlines settled into their present shapes, before forests spread across continents or ice gathered at the poles, the deep ocean already held its form, a vast, continuous environment extending through time with a stability that surface landscapes rarely maintain. Within that continuity, processes unfolded slowly, almost imperceptibly, guided by pressures and temperatures that have shifted only slightly across immense spans of geological history. The water at depth retains its cold, dense character, a few degrees above freezing, unlit and unaltered by the daily rhythms that shape the upper layers of the sea. Life emerged within this environment early, long before complex bodies or articulated movement appeared. Microbial communities established themselves around chemical gradients, drawing energy from the earth itself rather than from sunlight. Hydrothermal systems released heat and minerals into the surrounding water, sustaining ecosystems that have persisted, in one form or another, for billions of years. These structures, anchored to the seafloor, operate on timescales that stretch far beyond individual lifespans, maintaining continuity through gradual change rather than abrupt transformation. When the lineage that would become the sperm whale entered this domain, it encountered a world already layered with history. The descent from the surface into depth is not only a movement through space, but also a movement across time, each meter downward, bringing conditions that have remained consistent through eras in which continents shifted and climates oscillated above. The seafloor reflects that continuity, shaped by sedimentation and tectonic motion that proceed at rates measured in millimeters per year, preserving traces of events that predate vertebrate life. In this environment, the absence of light defines perception. Vision, dominant at the surface, becomes limited and eventually irrelevant as depth increases. Sound takes its place, not as a secondary sense, but as the primary medium through which structure is resolved. Each acoustic pulse travels outward, interacting with objects, surfaces, and organisms, then returns, carrying information encoded in timing and intensity. The result is not an image in the visual sense, but a spatial representation assembled from echoes, continuously updated as the animal moves. The whale operates within this acoustic field as a constant reference, its position defined relative to the returning signals that surround it. Objects are not seen but inferred, distances measured not by perspective but by delay. Movement alters the pattern of reflections, allowing the environment to be reconstructed moment by moment. This mode of perception is not intermittent but continuous, a steady exchange between emission and return that defines the boundaries of the whale's awareness. The seafloor, when reached by these signals, reflects them back from surfaces that have remained stable across vast intervals. Each echo carries information not only about present form, but about the persistence of that form through time. The contours that shape the returning sound were established long before the emergence of complex marine predators, before skeletal structures capable of supporting rapid movement, before sensory systems adapted to interpret such signals existed at all. Within this context, the whale's navigation becomes an interaction with an environment that holds memory in its structure rather than in transient events. The constancy of temperature, pressure, and darkness provides a backdrop against which movement occurs without altering the underlying conditions. The whale passes through, interpreting and responding, while the environment itself remains largely unchanged, a stable medium that records time through accumulation rather than fluctuation. What arises from this interaction is a form of engagement with the world that does not rely on illumination, color, or horizon, but on the precise interpretation of returning sound. The experience associated with this mode of perception cannot be directly compared to human sensory frameworks, which are anchored in vision and constrained by light. The acoustic domain operates under different principles, organizing space through timing and resonance rather than through reflected photons. The question of how this is experienced internally remains unresolved, not due to lack of observation, but due to the limits of interpretation. The structures involved are known, the signals can be measured, the behaviors can be described, yet the translation from these external markers to any internal state is not defined. The ocean, in its continuity, holds the interaction without revealing its subjective dimension, remaining present as both environment and witness, unchanged as the organism moves through it, leaving the deeper aspect of that experience beyond direct access. There is a form of understanding that settles comfortably, where each unknown appears as a temporary absence, a space that will eventually be filled as tools improve and observations accumulate. In that view, uncertainty is only a delay, and the structure of knowledge expands steadily outward, converting questions into answers with time. This model has carried science through centuries of discovery, providing a sense of continuity and direction that aligns with the steady refinement of method and measurement. Sperm whales do not sit easily within that frame. Their biology is not inaccessible, their behavior is not chaotic, and their environment is not beyond observation in any absolute sense. Yet the integration of what is known into a complete account does not resolve in the expected way. The elements remain clear in isolation, anatomy, acoustics, social organization, movement, each described with increasing precision, but the synthesis of these elements into a single, stable interpretation remains incomplete. This incompleteness does not arise from a lack of effort or a shortage of data. It reflects the structure of the questions themselves. Many of the tools developed within biology are optimized for systems where function can be inferred directly from structure, or where behavior can be mapped cleanly onto environmental variables. In organisms whose lives depend on internal states that are not externally visible, that mapping becomes less direct. The relationship between signal and meaning, between neural activity and experience, between social pattern and internal representation, introduces layers that are not easily reduced. The presence of a brain weighing approximately 20 pounds, organized with regions that have no clear counterparts in terrestrial mammals, indicates a level of processing that exceeds simple coordination or reflex. The acoustic environment in which this processing occurs is structured differently from the visual domain that shapes human perception, relying on timing, reflection, and continuous spatial updating rather than on illumination. Communication unfolds through patterned sequences that carry structure without yet yielding interpretation, suggesting organization without revealing its content. Within this configuration, the whale is not an unsolved problem in the conventional sense. A problem implies a defined question with a solution that can be reached through existing methods. What appears here is closer to a boundary, where the questions themselves extend beyond the range for which those methods were designed. The difficulty is not only in extracting information, but in recognizing what form that information might take once it is obtained. The scale of the environment reinforces this perspective. The ocean covers 71% of the planet's surface, extending to an average depth of approximately 12,100 feet, with vast regions remaining only partially mapped. Within that volume, conditions vary across gradients of temperature, pressure, and chemistry that operate independently of surface dynamics. The whale moves through this space continuously, its range encompassing regions that have remained stable across geological timescales, interacting with structures and processes that precede human observation by orders of magnitude. Across this expanse, the whale's activity persists without reference to human presence. Acoustic signals propagate through water, reflect, return, and are interpreted within a system that functions regardless of whether it is observed. Social interactions unfold, movements are coordinated, and behaviors are transmitted across generations within a framework that is internally coherent, even as it remains externally opaque. The existence of such a system does not diminish the capacity of science, but it expands the context in which that capacity operates. It indicates that there are domains where understanding will require not only more data, but also new ways of framing what counts as explanation. The absence of complete interpretation becomes part of the structure, marking where knowledge transitions into exploration rather than resolution. Beneath the surface, in regions defined by cold, darkness, and pressure, these processes continue uninterrupted. The whale moves, signals, and responds within an environment that does not announce its contents, maintaining a continuity that extends beyond immediate observation. What occurs there is present, structured, and real, even as its full description remains incomplete, a persistent reminder that the world contains layers of organization that are still in the process of being understood.